nucleotide excision repair. Four proteins are involved, that is UVR A, B, C and D. Normally UVR A detects the distortion along with the UVR B by scanning on the double helical DNA. Upon encountering the distortion, UVR A leaves, at the same time UVR B builds the double helical DNA by breaking down the hydrogen bond between two strands of the DNA. Further, UVRC is recruited by UVRP. It creates two incisions, 8 nucleotides away from the lesion at the 5' end, and at the 3' end nearly 4 to 5 nucleotides away from the 3' end. So, nearly about 12 to 13 nucleotides are long in the single stranded segment of DNA. Once the open complex is formed, that time UVRD shows helicase activity and DNA polymerase 1 fills the gap and DNA ligases it. Any mutation in various protein like XOA, XP, C, D, F and G, it produces 7 XP gene that resulting in affected individual, they are sensitive for the sunlight. We call it as xeroderma pigmentosum, that is a skin cancer seen in them. Double stranded break repair pathway. It retrieves the information from two sister chromatids. The recombination events are accompanied by a double-stranded break. Initially, when the break of two strands of DNA is associated, that involves DSB, that is double-stranded break repair pathway. Once the DNA cleaving enzyme is sequentially degrading the broken molecule, it produces single-stranded DNA on the tails. This single-stranded tail remains or terminates with 3' end as the 5' ends are degraded for both the strands. Then these single-stranded DNA undergoes strand invasion with the homologous chromosome or sister chromosome, which is complementary to each other, so that this invaded region then further acts upon the homologous chromosome, acts upon the strand that is responsible for the production of new DNA. Then further elongation of this DNA takes place using the complementary strand of the homologous chromosome. We call it as a branch migration. Once they are fused together, the two original duplex were identical in the sequence, then automatically the branched region, we call it as Holliday junction, could be lost during the recombination during this repair pathway. This non-reciprocal step in double-strand repair sometimes leaks a genetic trace, giving rise to gene conversion events. The two Holliday junctions are found in the recombinant intermediate generated by this model, that is non-crossover products and crossover products. Here we can see the difference between non-crossover and the crossover product of Holliday junction, which is found in replication. And further, the non-homologous end joining is also happening in the case of chromosome that suffers a break. Then no sister chromosome is present to serve as the template, the double-stranded break repair. So alternate system comes to play is nothing but non-homologous end joining. Further, this pathway is associated with various components such as Ku70, 80, DNA PK, Artemis XRCC4, Seminose XLF, DNA ligase, Ku70, etc. Ku70 and 80 are heterodimers that binds to the DNA to recruit a DNA PK, that is protein kinase. When the DNA PK binds to this complex with atremes, the 5'3' exonuclease, a latent endonuclease, it activates phosphorylation of both the strands by joining the broken end, and they prepare them for ligation process. Further, the ligation is in turn carried out by DNA ligase 4 with the help of XRCC4 and Semunose XLF. The two ends are then further joined by amalgamation. That broken ends are combined together during this non-homologous end joining process. Another one is translation synthesis. Translation repair, that is involve polymerases directly synthesize DNA across the site of damage, is a fail-safe mechanism. That is, along with UMUC and UMUD are the members of DNA polymerases. That they bind to DNA during replication process along with polymerase. That incorporates nucleotides that are independent of base pairing. Resulting in repair of the translations in the particular place. The final one is nothing but our SOS response, save our soul response. The protein called Lexia repressor or transcription repressor is active during this normal condition. It controls the expression of gene involved in repair such as UMUC, UMUD and UVRA. Upon damage, the damaged single-stranded DNA activates Recce protein that causes proteolytic cleavage of Lex-A, hence the repair system gets activated. So that's all about various repair mechanism. And further, P53 and P21 undergoes a multifunctional transcription factor that is encoded by P53 gene or tumor suppressor gene. It acts as a sensor of damage and it protects the cell, hence it is called as guardian of the genome. Under normal condition, it is negatively regulated by itself and it is present in stress amount because of its extremely unstable condition. During stress or hypoxic condition or DNA damage, nucleotide, the shortage of release of ATM and ERTR serine kinases, then post-transcription modification of P53 takes place. That P53 increases in concentration that pushes the cell to apoptosis or cell death. Here is a diagram that is associated with the action of P53 and P21 mediated cell cycle control. That's all about DNA repair mechanism. We'll see the other things in the next class. Thank you.